Petunias. Scientists wanted it darker, so they worked week after week and finally got... What happened? RNAi. Here, let me show you. Zooming into the flower petals, we see that it's made up of cells. In each one, the DNA coding safely locked in the nucleus is copied over into RNA sense strand, which hold the direct instructions that ribosomes use to manufacture these pigment-producing proteins, which make the flowers pink. The flow from genetic code to proteins is called the central dogma. It is key to every single living thing in existence. When the scientists added an extra pigment gene to make it darker, it turns out that instead of producing just the usual RNA sense strand, the additional gene produced both the RNA sense and antisense strand. What does that mean? Because they are sense and antisense, they pair up, but in this form, they look like viruses. When the cell detects these, a protein called dicer quickly cuts up the RNA into small fragments. Then the Argonaut family protein binds to these fragments and discards the sense strand. With the antisense strand, it joins other proteins to form the RNA silencing complex, or RISC. And once there is a match, the sizer protein cuts the RNA, rendering it useless. It even cuts the original ones too, destroying all pigment RNAs. No RNAs, no protein. No protein, no pigment. No pigment, completely colorless flower. But what can you do with it? With RNAi, we can target diseases like cancer by introducing paired up RNAs that code for its top lethal proteins. Slicer cuts all the RNAs that match, and because of that, no more lethal protein is produced, and thus we can quell these diseases or even eliminate them entirely. And the craziest part, we learned this big secret from something as simple as a petunia.